Hello everyone, this is the second lecture of our mind map series, and this is going to be the most important lecture of this unit, as it will be covering the this basic subunits up to 90%, and remaining two topics will be there. One is reproduction, and that is quite self-explanatory as we move through different phylums. The second topic which is there, that is related to classification with respect to evolutionary and developmental trends, that can be understood easily if these topics are clear. So this will also help in that. I will be making video on that as well if needed, but that can be done easily. The second reason being, I have compiled all these concepts in a single mind map, and you need not to refer so many sources or go through individual phylums to get all this information. And moreover, I have interlinked all the concepts so that it is easier to remember and recall, and for a much better understanding, this is being done at one place. So let's start our topic. The first topic which we have at our hand is this level of organization of tissues, means how tissues are formed, how different higher levels are formed. Then the first level is that of cellular. It's not the tissue level of organization. Cellular means tissues are not formed, cells are not coordinating with each other to form a specific tissue for a specific task. Here what is happening is that the cells are aggregating at a place and performing a certain function. The most important part here is that there is this concept of re and de differentiation, which you will not find in many books. This is the reason that the division of labor can be done. Here what happens that these cells are acting like the stem cells, so that they can perform different tasks and different functions as per the need of the cell. This is found in porifera, this cellular grade of organization. Now we are actually doing this from on the scale of non-chordates to chordates. This must be kept in mind. Now moving to this tissue level of organization, here what happens is that uh, the cells are coordinating with each other, they are coming together and forming a specific tissue for a specific task. For example, nerve net in coelentrates. This is found in coelentrates. Then let's come to organ level. In organ level, various tissues are coming together and coordinating with each other to form an organ. And this organ is having two parts. One is this parenchyma part, which is functional part. Second is the stroma part, which is actually the bedding. This parenchyma can be formed of various different tissues, not only a single kind of tissue, to perform different functions, because an organ can perform different tasks related to a common thing, like reproductive organ. It is having a specific task, but in that specific task also means formation of gametes, various tissues are performing their various functions for that. It is found in platyhelminths. Now let's come to organ system level of organization. Here simply various organs come together, they coordinate to form a system which is known as organ system. It is in the first phylum where it is found in, it is there in escalmins, in the nemerton worms. Example is reproductive system, digestive system. Like here there was reproductive organ, but various reproductive organs come together to form a whole system by the coordination and cooperation of different organs. This way things become more complex but more sophisticated also. It is found in these groups: escarpments, annelids, arthropods, mollusks, echinoderms, and all the way up to the vertebrates. Now I have segregated these into two groups due to a reason which will be clear to you in the later part of this video, but these two groupings are important here. Now let's come to our second topic, which is symmetry. what is symmetry? The symmetry is a process where, or we can say a phenomena where uh, an organism can be cut into similar parts or wholes, known as enteromeres. They are similar in their arrangement of the various organs or tissues or parts, we can say. It is known as symmetry, and the organism which is showing some symmetry is known as symmetrical. The organism which is completely not showing any kind of symmetry, it is known as asymmetrical. For example, now let's come to the interlinkage also. In porifera, we can see that there is no symmetry. Mostly they are asymmetrical, except Sycon and Leucosolenia, which are having radial symmetry. We will discuss what radial symmetry is, but they are mostly asymmetrical. Now let's come to radial symmetry. In radial symmetry, what happens that there are two conditions. First is the central plane and second is top to bottom. See, this is the central plane. These are two central planes which are passing from the center of the body, but from top to bottom. If these conditions are fulfilled, that a central plane cutting from top to bottom can easily cut an organism into different enteromeres which are similar, then the symmetry is radial. It helps in food detection and detection of danger because 360-degree stimulation can be there. So there are various cases like biradial, means two planes, like here we can see two planes can cut an organism. There can also be a pentaradial, means five such planes are there which can cut the organism into five or six enteromeres. Five planes and six enteromeres. It is found in echinoderms. It is an exceptional case which we will discuss. Now let's come to interlinkage. All the organisms which are having tissue grade of organization, for example coelentrates, they are having radial symmetry, except this echinoderms, because these are not having tissue grade, these are having organ system grade, as we can see here. This is the exception. Now let's come to bilateral symmetry. Bilateral symmetry is that an organism can be cut into two halves only by a single plane, which is anterior posterior plane. Like see here, it is a single anterior posterior plane cutting from the midline of the body, and it is cutting the organism into two equal halves, this one and this one, left right kind of thing. This dorsoventral plane can also be there, but it is not cutting the organism into two similar halves. Both are different. Now where is it applicable? It helps in civilization. Civilization means formation of head. But why is it important? Why has this term been given? Civilization is that various nervous tissues are getting accumulated at a particular region, and various sensory organs like eyes, tentacles, and various antenna in the insects, they get accumulated at a particular region which is known as head, or and this process is known as civilization. This helps in movement of an organism in this forward direction. This is important. Now let's come to interlinkages. We can see all the organisms with organ and organ system grade of organization, they are bilateral in symmetry, except we know echinoderms. In echinoderms also the larval forms are bilaterally symmetrical. So here you need not to remember each and every phylum now for bilateral symmetry and just keep it there in the memory. Just you have to be knowing what are the phylum where we can find organ and organ system level of organization. There only we will be finding this bilateral symmetry. These are the phylums. This is there. Now let's come to our third topic, that is the tissue layers. Here it is, symmetry and tissue layers. The first category is no tissue. We can see that in porifera there are no tissues. So all the organisms having cellular grade of organization are asymmetrical, except these two are having no tissues. They are having cellular grade of organization. And one thing which is important here is that we can only find blastula stage here, no gastrula like this. And why is it important to study these embryological uh, figures or these details? Because this way we can easily get to know why is a particular organism having different kinds of layers and what is the interlinkage. And it will also help us in understanding the formation of coelom, which is our next topic, and then in classification also. And it will be easy to remember and it will be a broader understanding. That's why this embryological thing is important here. So no gastrula is found in porifera, only blastula. This is blastocele, and that's why only cellular grade of organization is there. Now, in case of diploblastic organisms, which means two layers are there, diplo means two, what happens that there is the formation of gastrula, which leads to formation of two layers. Here from blastula itself gastrula is formed. In blastula, here we can find there is a blastopore. From this blastopore there is invagination, and it leads to formation of this cavity, which is known as archentron cavity, which will be forming gut in later parts of the development. Then we are having this blastocele, which is another cavity. But due to this invagination here, again a layer is formed. First there was only this thing, which is ectoderm, but due to this invagination there is this formation of cavity and this layer known as endoderm. Now we are having two cavities, endoderm and ectoderm. Sorry, two layers, endoderm and ectoderm. That's why diploblastic. And two cavities, blastocele and archentron. This is the blastopore region. Now let's come to interlinkages. All the organisms having tissue grade of organization are radially symmetrical, except this are diploblastic. This is a universal thing, except this, because this is a triploblastic, as we will see. Now both these figures are similar, quite similar, just one difference is there. Here is a bunch of cells which have actually been formed by this endoderm, and these are the mesodermal cells. They just fill this cavity, which is known as blastocele, and they form a third layer, which is known as mesoderm. Thus three layers are there: endoderm, mesoderm and ectoderm. That's why triploblastic thing is there. This is same gut cavity archentron. This is blastocele. See here, these are the three layers. This pink one is the mesoderm, this is ectoderm, this is endoderm, this is the archentron which has formed the gut, and this blastocele has been completely filled by this mesoderm layer. Now let's come to interlinkage. All the organisms having organ and organ system grade of organization are having bilateral symmetry, except this are triploblastic. Just this is the interlinkage. We can also see that all the bilateral organisms are triploblastic. This is the interlinkage. Now let's come to our last topic, coelom and its types. First we must be knowing what a coelom is. So here is a basic universal definition. Coelom is a body cavity between ectoderm and endoderm. See, between is this a body cavity between ectoderm and between endoderm. We can also say that between the body layer and the endoderm means gut. See, here is gut, means the coelom is a body cavity this between this ectoderm or body layer, and the endoderm, or we can say this gut cavity, lined by mesoderm on all sides. This pink is the mesoderm, and it is lining this cavity from all the sides. This is the concept of coelom, and these are the mandatory conditions, that it has to be bound by mesoderm and on all sides, and it lies between this ectoderm and endoderm, or body wall and gut. This is the definition of coelom. Sorry, this is not leaved, this is lined. Now let's come to various concepts of it, and then we will move to interlinkages. Acoelomate means the organisms which are lacking this body cavity. First organisms which are acoelomates are porifera, because they are having tissue grade of organization. There is no cellular grade of organization, there is no tissue. That's why simply they are acoelomates, because if tissues are not there, then there will be no cavity. Then let's come to coelenterata. In coelenterata, what happens here, we can see that there are only two tissue layers, no third mesodermal layer is there. So if there is no mesoderm, what we saw here, that it has to be lined by mesoderm on all its sides, means even if a cavity is there, right, even if a cavity is here, it will not be a coelomic cavity, because it is not lined by mesoderm. We will see that case also. Then let's come to platyhelminths. Like here we can see that this blastocele cavity is completely filled by this mesodermal layer, and there is no cavity. They are acoelomates. So the interlinkage, let's come to first interlinkage up to this part. This is an exceptional thing. All the organisms having cellular and tissue grade of organization are radial or asymmetrical as per their grade, and all the diploblastic are acoelomates. The only triploblastic which is acoelomate is this platyhelminths, because mesoderm is there. Triploblastic means mesoderm is there, but there is no such cavity. Now we can see that the organ level grade of organization is also ending up in this acoelomate category. The only organization where we can find this coelomate condition is this organ system here in these groups. And now you will understand why I have split these into these two groups. Just keep this aside, this is a special case, S. helminths. Take this group, Annelida, Arthropoda and Mollusca, and then echinoderms to vertebrates. We will see here now the concept. What happens, like I told you, that this mesodermal layer fills this whole cavity. From here also the whole cavity gets filled. Then the first case is schizocoelomatous condition. What happens here is that this filled tissue means the mesodermal tissue which gets filled here it is being split, and the process of splitting is by programmed cell death, by apoptosis. There is splitting of these mesodermal bands, and there is the formation of cavity which elongates and fills this whole blastocele and forms a coelomic cavity. See here, there is no blastocele now, and again if we see the definition of coelom, the conditions are getting fulfilled. Like how coelom is a body cavity, this which is completely lined by mesoderm from all the sides, this, right, both the chambers, and it is lying between this ectoderm and endoderm, or we can say body wall and gut. The conditions are getting fulfilled and blastocele is completely gone. This condition is found in annelids, arthropods and mollusks. Here, see this group, annelids, arthropods and mollusks. This is a special case, just leave it aside. Then there is a second group from echinoderms right up to the vertebrates. There what happens, there is another, means they are also coelomatous, but the formation of coelom is by a different process. Here it was by splitting of these mesodermal bands, but here what happens right from the start, during the gastrula phase, various mesodermal cells arise from the gut. They form these pouches, mesodermal pouches, and these pouches then bud off into this cavity, right? See here, there is no such splitting, just these pouches have bud off. Now they elongate and fill this whole cavity, and again we are having a coelom between ectoderm and endoderm and lined completely by mesoderm, and blastocele is completely gone. This is from echinoderms to higher vertebrates. Now coming to special category of S. helminths. What happens here? In S. helminths, there is this concept of pseudocoelom. Why? You can see here, this is a body cavity between ectoderm and endoderm, lined by mesoderm, but only on this outer side, not the inner side. On the inner side, the blastocele has fused, actually. It means it is the blastocele actually which has given rise to this cavity, and mesoderm is only on this outer side. This is found in roundworms, and this is a pseudocoelomatous condition. Right, now let's come to the interlinkage, means all the organisms having organ system grade of organization, these two groups, keep them in mind, are bilateral in symmetry, are triploblastic, and are coelomatous. And there are two groups, schizocilous and enterocilous coelomatic conditions, right? And we had earlier discussed these interlinkages also. Now let's quickly come to the benefits or significance of this coelom. First, there is organ development. Just mix it with this thing, this point. This is important. Organ development, various organs develop from the mesoderm, and they like reproductive organs are there, and they are also this space, this cavity is also providing space for organ growth. One important thing is that in invertebrates, this layer is known as mesoderm is, this is a mesodermal layer, but in vertebrates this is known as peritoneal layer or peritoneum, and this cavity is known as peritoneal cavity. There it is a fluid filled cavity, right? And in case of invertebrates, in case of lower organisms, this is not known as peritoneum, this is known as mesothelium, and it is there also, it is fluid filled cavity. So in lower invertebrates like annelids, it is also providing a hydraulic skeleton for movement, for diffusion of nutrients and waste materials. It is also helping, and for protection of various organs, as it is providing a cushioning effect. Like there is this pericardial cavity which is providing cushioning for the heart, and likewise there can be many other benefits also, but these are the most important and significant ones. So with this we completed our lecture. I hope that this has benefited you, and this will benefit you once you revise this well and you make all those interlinkages and remember these, so that the whole picture gets clear, and for the next topic also it becomes easier, means that classification kind of thing, that becomes very much easier once we go through this. This is actually kind of bedrock for that topic. So keep revising, keep learning, and keep supporting us. Do subscribe and join our Telegram channel also for more such videos and stuff. And also, if you find our test series beneficial as we have made that meticulously exactly as per the demand of the exam and exactly as per the topics of the syllabus, and we have made it much more affordable to all the students. So do join that through our Telegram channel. And with this we end up here. We will meet for our next lecture. Till then, goodbye. Thank you.